there's some issue with. Switched off the camera just to tackle the network a little better. Okay, so this should be fine now. Okay, so let me just try again. Yeah. So hello everyone, welcome to week 4 of our classes for genes, genome, and the epigenome. As you might recall, previously we have been discussing a number of different topics. We have majorly, let me just go back to our previous class because that was a doubt that somebody had. So I think, yeah, somewhere here we had discussed. Okay. So, very initially, we had discussed topics which were centered around the discovery of DNA, the different molecular biology experiments that helped in understanding the same, including those which were conducted by Hershey Chase and Frederick Meister, and the conclusion that DNA is indeed the macromolecule of the cell, which helps in passing on information from one generation to the next, and that the integrity of that information is maintained because of the chemical nature or the structure and the architecture of DNA as a hereditary molecule. And further on, we understood how the genome is organized, how genes are present or they are aligned within the entire stretch of the genome. What are the different components? What are the coding regions and the non-coding regions, and how do they make a difference? By difference, I mean that the positioning of a gene, what elements are present close to it, whether there's an enhancer, whether there's an insulator, um, yeah, whether there are any other regulatory elements that actually help in understanding the same. All of that information was previously discussed. We talked about regulation of gene function. We talked about regulation at an epigenomic level. However, we did not get into much detail about it because of these topics will be covered later on. And then we moved on to understanding how DNA methylation has a role to play in context of gene expression. Let me just open up the slide. I think it'll make it easier. Yeah. So here we talked about how DNA methylation has a role to play, where it is present and which elements are getting methylated, whether or not, like it's not an absolute 0 or 1 in terms of controlling or regulating gene expression, but instead it is the stretch, it is the presence of the methyl group over a stretch of nucleotides or a specific region of the genome that can actually control transcriptional repression or some other activity. So we also discussed the different enzymes that have a role to play in it, the regulatory molecules like or regulatory proteins which often bind to these methylated sites and then control how gene regulation will actually be facilitated, and some of the diseases that are associated with improper or incorrect methylation were also mentioned, like for example Prader-Willi syndrome or these diseases like Angelman syndrome, etc. So these are all the different aspects that were previously discussed to understand how gene expression is regulated at multiple levels and how DNA methylation in particular has a major role to play in this context. Later on, we also understood how in context of cancer, DNA methylation particularly is very important. Like for example, how in a normal cell, there are stretches of the genome that are necessary to be maintained under hypermethylated conditions, but in a neoplastic cell or in a cancerous cell, this often changes due to a number of different reasons, and which type of gene is subject to this change will actually determine whether or not that event can be an oncogenic event or not, or whether at all it will not have any effect within the cell. All of these differences can be understood. Later on, we also understood the role of mutations and how that actually has a role to play in wreaking havoc in the genome. The different types of mutations, whether they are induced by some sort of a chemical entity or by some other reagent, or if it is spontaneous in nature, it is arising just because of the partial fidelity or infidelity of an enzyme which is supposed to do its job perfectly under normal circumstances. When that gets disrupted, then it can wreak havoc in the genome. Then we understood the genetic code, and this was necessary because it actually helps in understanding mutations a lot better. I'll not go into details of this because it was already discussed. Then we understood the different types and subtypes like point mutations, silent mutations, nonsense mutations, the different or rather the practical implication of the presence of such mutations. Um, just a second. Let me. Yeah. So the implications of such mutations, and later on we understood indels or insertion and deletion mutations and their role in context of there being a shift in the reading frame, which is understood or which is interpreted by the ribosome at the time of translation, whether it is in frame or whether it is leading to the either a change in the amino acid sequence or it is leading to a nonsense codon. All of these possibilities were discussed. Then we understood other subtypes of mutations like neutral mutations, silent mutations, etc. And this was a summary of what was previously discussed. Then, sorry. Then we understood how DNA replication errors have a role to play in this context, which is a subtype of the spontaneous mutations that I just discussed, where it involves the shift or um, the tautomeric shift or the existence of a chemical base in different orientations or in different states, which completely alters the type of base pairing it can form. And as a result, it effectively leads to a change from one type of a base pair to another type, which will initially, if detected as a mismatch repair, will be taken care of. However, when there is a large number of such mutations occurring at different points within the genome at the same time, then for the cell it becomes too big a burden. And under those circumstances, the accumulation of such DNA damage or rather mismatch repair defects eventually can lead to cancer. And it is a very common subtype, which is not even restricted to one cancer type, but it is abundant in different oncogenicities as well. Just for a second, I think it got minimized. So then we talked about spontaneous chemical changes like depurination or deamination and their role, how they can affect the chemical nature of the bases that we are studying, and that can, in subsequent rounds, also lead to either a defect in the ability of the enzyme complex to move. Like for example, if it is a thymine-thymine base pair which is forming, that is a thymine dimer which is forming, then under those circumstances, it is creating a physical obstruction or a structural burden on the cell. And as a result, when such a huge obstruction comes in the way of the machinery which is trying to read that region of the DNA, then the replication machinery pauses. And as a result, such stuck replication is also very traumatic for the cell, and such a cell can also face a lot of trouble when there is repeated accumulation of such changes. Then we talked about base analogs, like for example, 5-bromouracil and how its altered base pairing capabilities can also cause a change. We have discussed this extensively, so I'll move on from there. Then we talked about different agents which are intercalating in nature. The most common example everyone is usually familiar with involves ETBr or ethidium bromide, but there are other reagents as well. But what they effectively end up doing is causing mutations by the random addition or the random deletion of certain bases, which completely alters the base sequence in subsequent generations. Then, then we moved on to topics for week 3, where we understood the differences between mutations and polymorphism. So I think somebody had asked to discuss this one more time. So we'll go over this in a little more detail. So the key difference arises between the key difference that we talk about when we say a mutation versus a polymorphism is the fact that a polymorphism by default under normal condition is a simple variation in the DNA sequence which is present in the population. So if you look at the key definition, then it is simply called as a variation in the DNA sequence which occurs in at least 1% of the population, maybe even more. Now, under the banner of genetic polymorphisms, you have multiple subtypes, like there are single nucleotide polymorphisms or SNPs, which is what we commonly discuss. Then you have microsatellites, you have small insertions, small deletions. So they all come under the same banner, but whether or not they can be disease causing or not disease causing is context dependent. Let us first look at the contrast, which is shown on the right. So you have mutation versus polymorphism. The mutation is a permanent alteration of a nucleotide sequence of a gene. Polymorphism is the presence of one or more than one, not more, it's usually more than one allele at a particular locus in a particular population. So the definition of polymorphism is not with respect to one individual, it is not with respect to even a set of individuals, it's with respect to a large number of people. And among those people, when you look at the sequence of a certain region within those n number of people, say for example all the Indian males, if you're taking them into consideration and then looking at their genome and then looking at some gene X within their genome, and then you are screening for similarities or differences within their entire genome sequence, then that will give you an idea of the polymorphism which persists. In contrast, when you talk about mutation, that arises from a different background, based on what I just discussed previously, that it can either be a spontaneous event where polymerase errors have incorporated that change, or it can sometimes be some sort of a chemical entity like or even ionizing radiation like X-rays. All of these events are causing a change in the sequence of the genome, and that is a single base pair change. It is usually associated with disorders. Mutations are associated with disorders like sickle cell anemia, hemophilia, etc. On the other hand, when we talk about polymorphism, it simply shows you variety within a population. The key example are with respect to ABO blood grouping. So that is a result of polymorphism which persists because it's not leading to a disease condition, but it is leading to more variety. Just pause. I think it's otherwise the videos are too heavy to upload. So I'm just switching off my video for now. So yeah, so these were the major contrasts. And if you look at the types of single nucleotide polymorphisms, which is shown here in the figure, you will see the different varieties which are defined based on where they are present and what is their impact. So SNPs or single nucleotide polymorphisms are sequence variations where one nucleotide gets substituted for another. Now, you might say that this is a commonality between both polymorphism and mutation, but like I said, mutation is the result of some sort of an external agent or some sort of an error, but polymorphism is simply that persists by default within that population. So the effect of a SNP can or cannot affect the encoded protein, but most of the time it does not have any deleterious or, for that matter, any effect. It just leads to more variety. SNPs of medical significance, they cause a substitution in the regulatory or the coding regions that affects the expression of function of the protein. So if it is a SNP, okay, let's first look here. Most commonly, what we are interested in when we talk about a disease, when we look at SNPs or SNPs for a particular disease, again, the most common example being cancer, then we look for SNPs present within the coding region. Why? Because you expect that to have a change or manifest some sort of a irregularity in the encoded protein. So these occur in the coding region. They affect the protein function and they most commonly changes amino acid sequence. However, that's not the only type. You can have non-coding SNPs which occur in the regulatory region of the gene, like for example, the promoter, the enhancer, insulator, etc. And that will control how much of the protein is produced and how it can respond to some sort of an external stress. So, or external factor. So what do we mean by that? Say, for example, if the expression of a certain protein, for example, heat shock proteins, the most ideal example for such circumstances, it is usually dependent on recognizing the signal that the cell is surrounded in an environment where it is subject to extreme heat, and therefore to withstand that temperature change or that environmental change, the cell will express heat shock proteins, and therefore it will try to salvage or will try to survive. Now if that stimulus is no longer getting recognized, or if the gene respond or if the gene is not sensitive to that recognition anymore, then that can produce a deleterious effect. So if there is a SNP which is present in that region, in the regulatory region, it will be called as a non-coding SNP. There are also silent coding SNPs which are present in the coding region, but they are considered to be silent because they do not alter the protein's amino acid sequence or function. So they are simply there, but they're not causing any major defect. So this is actually quite common. They are mostly in any population you see. You will find like a few SNPs or a single nucleotide changes among different individuals, but that does not usually manifest into anything else, any disease or any worrisome conditions. Now, how do we understand the relevance of such SNPs or such genetic polymorphisms? Most common example that I've mentioned here as well is with respect to the response of different individuals carrying different SNPs, and their ability or their response to any sort of a drug uptake is a major example of the same. What do I mean by that? So if you look here, genetic polymorphisms can influence the pharmacokinetic and pharmacodynamic, uh, not abilities, but the way in which a drug is. Say, for example, you and I, we both are taking a certain medicine for cold. However, the two of us do not necessarily have to or will respond to the drug in the same manner. We might see that one of us absorbs the drug better, has better metabolism. Some of us might completely flush it out or excrete it out, and as a result, the drug is not having any effect. But the other one might be very responsive. Pharmacodynamic in the sense that the responses to that drug uptake that can be quite different. Say, for example, we have receptors which like ABC transporters or other receptors which are insensitive to the drug binding, or we have mutated as a different example, but they are insensitive or less sensitive to the drug, etc. So there are these variations which are present at a molecular level, and they are the consequence of single base changes, which are genetic polymorphisms. But the effect or the phenotypic result of those changes is the fact that we respond differently to the same drug uptake. So that was just one example. Then we also saw here that if you take an individual A and an individual B, and as you can see, most of their sequences, this is assume any region X within their genome from position X to position Y. And within that position, you can see there's only a single base change, which is called as a SNP. And when you look at the haplotype of these individuals, like all these set of alleles, then you will see that SNP1, SNP2, and SNP3 might be present overall. Now, it may be possible that the presence of a combination or like for example the presence of one SNP or a combination of certain SNPs are actually determining this drug response activity that I just talked about. Say individual A, B, and C, among them, if you have an individual A and they have the set of SNPs in this manner, this is just a representation of the same, that they have certain SNPs at certain regions, that allows them to be a very fast responder, and therefore higher doses of drugs might be needed to be provided. So this is just an example. Somebody might have no response, somebody might have high response, somebody might find very basal level of a drug to be very toxic, whereas the other one is not responding to basal level and needs very higher doses. So all of this, it cannot be ascertained by looking at a person. You will have to first of all sequence that person's genome, and secondly you will need to have a study where you know that the presence of X polymorphism or Y polymorphism is associated with a certain phenotype, and even then it would not be completely foolproof. So therefore, practically what doctors do is they start with a basal dosage for everybody, and then they ask the person or the patient to concerned to keep an eye on themselves and see how they respond to taking that drug. If somebody is responding very well to a lower dose, they will not be unnecessarily subject to a higher dose, and vice versa. So that's just a simple example. Also, SNPs in transcription factor binding sites or TFBS affect the binding to gene promoter regions. SNPs in the Tata box, they affect promoter activity, whereby A to C substitutions decrease the number of Tata boxes, and they decrease methylation. They affect non polymorphic CPG and transcription factor binding. So this is just an example of how SNPs present in TFBS can actually make a difference. So, for example, like you can see, if there is a SNP which is present within the transcription factor binding site and its ability can be controlled, then promoter activity will be influenced. Same from the Tata box. If it is in a CPG island region, CPG island is simply dinucleotide stretches of CG repeats. And as a result, these are more prone to methylation. And you can see the effect of SNPs in such regions based on how extensive that methylation within that region is, and therefore how well regulated it can be. So these are again simply just some examples. Um, these were more detailed examples. I'll not repeat the entire topic again, but basically what we see that SNPs present in different regions can have different effects. Like for example, in non-synonymous exonal SNPs, which means that these are SNPs which are present in the exon, which is the actual region which is getting encoded. What will that do? That will change the amino acid sequence of the protein, and therefore it can have potentially harmful effects. Intronal SNPs can not directly affect the protein's functional ability, but they can affect, influence or act. They are able to influence or act upon the gene expression or regulate the gene expression through cis-acting regulatory elements, and that is simply another example. Then we went into some detail about functional genomics. I'm not going into details of this, but different aspects of functional genomics. And then we finally moved on to sequencing techniques. So before I move further, if you have any questions regarding the SNPs part or any of the previous topics, please let me know. And then we'll move on to sequencing techniques, which is what we are taking up in more detail today.
No, ma'am, I have only doubt in that portion, so it is clear.
Oh, it's clear? Okay. So, for sequencing techniques, we had started off in the previous class with Sanger sequencing. So, if you remember, I'll just quickly like brush up the basics of this technique. So, first of all, what is the idea? Why are we spending crores and crores and unimaginable amount of money on sequencing? That is because it is a starting ground for us to learn about the genome, because the genome holds a lot of secrets that we don't understand even today. And when we talk about sequencing, it's not only restricted to the genome, we also understand the transcriptome, we understand the cystrome, which is the regulation within the genome. And we will discuss all of these techniques, and they are very cool, very cutting edge techniques, which are extensively being used in today's time. We understand the 3D architecture of the genome. All of this is understood simply because people learned how we can actually know the sequence. So the idea started small, but it is immense. It has unraveled, it has opened up biology to unimaginable prospects. And therefore, knowing basics of the techniques, even if not in extensive details, but at least having the idea of what it is, will really help not just you, not just me, but anybody who wants to work in the field of biology, because it will become almost impossible to avoid learning about these techniques. And you don't have to be scared. I'm also not from a bioinformatics background. It doesn't matter that much. You need to understand the basics, and you should be open to learning. That's all you need to do. And rest, there are so many tools available, there are so many softwares which are open access, there are extremely cooperative people who have built these softwares, who have established the codes which people use for analysis, and they're very approachable. So it's this domain, while it might seem scary to someone, it's necessary, and it is a huge achievement for mankind, so we should learn about it. But before we move into these cutting edge technologies, let's learn about the basic. So when we started off in the last class, we discussed the Sanger sequencing method, which is also known as the dideoxy chain termination method. And also we talked about Maxim Gilbert as well. So just a second. Yeah. I'll just come to the Sanger method directly. Maxim Gilbert we had already discussed in the last class. But basic idea of Sanger was to use a combination of dideoxy and a dideoxy nucleotide triphosphates and regular DNTPs, which are deoxy nucleotide triphosphates. And at the time of when you're providing primers, then at the time of amplification, whenever a complementary nucleotide bearing the dideoxy group is incorporated, chain termination or the synthesis will terminate at that point, and as a result, you will be able to decode which is the base which is present in that position. And this has to be repeated over many, many, many rounds to be able to decode the entire sequence. And initially, while the way in which these results were visualized involved the use of radioisotopes, where radio-labeled nucleotide triphosphates were used. In today's time, we use fluorophores, of course, because that's less toxic, not lethal, and it has become very affordable in the recent past. So currently, Sanger sequencing is utilized. I think I mentioned this in the last class as well. It is utilized only or majorly in context when you are trying to confirm a cloning that you have performed. So, for example, when we synthesize or when we use some sort of side-directed mutagenesis strategy using a primer or something, and we clone it into a plasmid, then we want to make sure that our sequence of interest is actually present in the plasmid. Then we usually opt for Sanger sequencing. It's a short method, and its limitation mainly lies in the fact that its um, fidelity is not very high. However, it has increased incredibly in the past few decades. I think initially Sanger sequencing could be done for 50 or 60 base pairs, after which enzymes would not be able to proceed further. However, in today's time, we can go up to, like most companies will offer you up to 1 kb, but good results are usually observed up to 600 or 700 base pairs. So that has been an immense feat or an immense contribution to the field of biology. So that was the basic concept. And this was the comparison between then and now, where you can see fluorescently labeled DNTPs were used or fluorescently labeled DNA sample was used. Either of the two strategies were used. And based on the length and the order in which each of the bases were getting recognized, you can see that the peaks present here are representative of the chromatogram of the sequence that you have um, or of the bases that have undergone sequencing, and from this you can decode the sequence. So that was the idea. And in today's class, what we are going to majorly aim at, let me just go back. Yeah. So if you just take a quick look at the overall content of the module, we are currently here, introduction to sequencing methods. So we are pretty much on time. So don't worry about it. It's a long course and you'll learn sometimes very diverse things. But for now, we are going to go into a lot of depth about next generation sequencing, which will not end in today's class. I will be focusing on a portion of it, a large portion of it today. And hopefully the rest should be completed by our next class, which is on Friday. And after that, we'll move on to more details about genome exome and transcriptome based techniques that we have just touched upon in our previous lectures. So the question that I'm starting off with is, what do we mean by next generation sequencing, or NGS, as you might very commonly have heard of it? So NGS, or next generation sequencing, is a massively parallel sequencing technology which offers you ultra high throughput, scalability, and speed. The technology, which is used to determine the order of nucleotides in entire genomes, or some targeted some targeted or some specific regions of DNA or RNA. So your starting molecule or your starting sample that you provide to any service provider, any company, is either your DNA sample or your RNA sample, which you ship to them. Then they carry out some quality checks. By quality checks, they basically ascertain that what they are using as a sample for your sequencing is good quality, is reliable. Reliable in the sense that when you subject it to sequencing, it doesn't give you incorrect or false results, and then they proceed with it. Um, what is the idea or the application of NGS? So this is overall. Let me just read it. It has fundamentally changed the kind of questions that scientists can ask and answer. So NGS allows labs to first rapidly sequence whole genomes. Second, deeply sequence target regions. What do we mean by deeply sequence? It means get more information, or you invest more computational power to no, sorry, not invest computational power, but rather do more iterations of sequencing for some specific regions, specifically amplify certain regions of the genome, and then you can compare it between different samples. That is deep sequencing. I don't think we're covering it in this class, hopefully in the next, but deep sequencing and long read sequencing we'll do in the next class. Utilize RNA-seq or RNA sequencing to discover novel variants and let's yeah. Utilize RNA sequencing and discover novel variants or splice sites and quantify them for gene expression analysis. Analyze epigenetic factors such as genome-wide DNA methylation, DNA-protein interactions, and chromatin accessibility. Sequence cancer samples to study some rare somatic variants, tumor subclones, study human microbiome, and identify novel pathogens. These are all the possible functions. Of course, there's more to it. Mainly because people have now shifted their focus from this from the idea of only getting the DNA sequence to understanding the architecture of the genome. Mainly, we now look at techniques like ChIP-seq, we look at ATAC-seq, we look at HiC, all of which I'll be discussing later on in I think maybe the 10th or maybe the 12th or 14th week, somewhere around that time, we'll be discussing those topics. They're also very new, very relevant, and extremely informative techniques, and they give us so much more insight into how cells actually function. So, coming back, what we see here is a comparison between Sanger sequencing that I mentioned just previously and NGS or next generation sequencing. So, let me just zoom in a little bit. So what you see here are the starting materials. So you have nucleotides and you have template for Sanger sequencing. And for Sanger, you will have one single template. For NGS, this first step is the main difference between both the techniques. Millions of templates are present for NGS studies. How do we get them? For to ascertain that, because that actually is what defines the word high throughput. High throughput means that you will initially use very limited amount of sample, but the processing that it undergoes is huge. Okay. And the outcome that you get from the same will also be very extensive. So it is giving you a large amount of data when a very small amount of sample or very small amount of initial product has been utilized. So that is the idea. Just give me a second. Just let me know if you can hear me. I just switched my, if you can't hear me, let me know. There was some issue in my audio. So, yeah, that was the idea behind, or the key difference between Sanger and NGS. And also, there's a mixture of bases. We'll come to it later, but this is the key difference between both the techniques. Next, what you see is for Sanger sequencing, you have extension and you have arrangement. So, like you can see, you will have hundreds of such nucleotide stretches or nucleotide sequences. And based on the length, like for example, you will have A, T, G, C, T, A, G, C, T, G, A, and T. So in this manner, you will know that this is the sequence that it reads from 5' to 3', and then you will be able to decode that based on your chromatogram as well, because the shorter sequence will appear first, the longer sequence will appear later, and that's how you will know that this is the sequence that I'm getting. In contrast, the steps which are a part of next generation sequencing are quite different. Um, don't worry too much about it. I have multiple slides that will discuss these in more detail. But overall, after generating millions and millions of templates, you will have to go through some sub steps, which includes arrangement, extension, detection, and restoration. And this particular these particular steps are part of a loop which is repeated more than 100 times, where extension, detection, and restoration are carried out, and then array imaging is performed, and then the sequence is decoded. That's all you can just focus on for now. Then, where did it go? Just a second. Yeah. Now we'll focus on some key concepts of next generation sequencing. Overall, the entire, okay, if you have queries, if you can't understand anything at any point, please don't hesitate. Please interrupt me and we'll discuss these. These are possibly new topics for students, and it can be confusing. So we'll take the discussion very slow. So the entire NGS workflow is majorly divided into 4 steps. What are those? First, you have sample extraction from whatever cell or tissue that you're trying to study. Yeah. So sample extraction is performed whether you're carrying out RNA sequencing, then you'll have to isolate the RNA if it is exome, or if it is DNA, then you'll have to whole genome sequencing, then you'll have to isolate the DNA. You can also sometimes ship the entire tissue sample. Say, for example, if you work with patient tissue, then the patient's surgically excised tissue can be directly shipped to the service providers, and then they carry out the entire sequencing process and they simply share results with you. So with high throughput shotgun Sanger sequencing, genomic DNA is fragmented. Then it is cloned to a plasmid vector and is used to transform E. coli. What does this mean? The first step that you see here is not the first step, but the first key terminology that you see here is the use of shotgun Sanger sequencing. What is the meaning of this shotgun that they're talking about? So basically, the idea is that it's similar to an explosion, which is the result of shotgun or a gunshot. Similar to that, genomic DNA undergoes fragmentation. How does it undergo fragmentation? So, for example, just let me switch on the video. Help me explain it better. Yeah. So for any of these techniques, if you consider cells, then you will see that you have, say, for example, 5 million cells that you are extracting your DNA or your RNA from. Let's talk about DNA only for now because it's easier to interpret and easier to understand. So if you have long stretches of DNA, which is how eukaryotic DNA usually is, then sequencing that, first of all, will be very difficult because the primer will find it very difficult to go and bind to its sequence of interest. And secondly, because DNA is not present, in a beautifully organized manner, like in a stacked manner or anything of that sort. No, it is completely packaged into chromatin, like we have discussed previously. It is not accessible, and secondly, the entropy of the primers will be very restricted because they will physically find a lot of hindrance to go and bind. So as a result, what is extremely important is to break down the DNA, not to chop it. You don't want to use an exonuclease and digest DNA into nucleotides. No, you want decent sized fragments like 100, 150, 200 base pairs, or sometimes even longer, which can be then individually taken at a time. And then the results from all of those nucleotides can all be pooled. And then the results which you get can be used for conclusion. So to facilitate this, what is usually done is that you get commercially available kits to isolate DNA and RNA. Again, let's talk about DNA only. You can use those kits. You can use traditional methods. You can isolate complete DNA from a cell, and then you can subject it to sonication. So, what sonicators do in the current scenario, they are capable of actually um, breaking down DNA to certain lengths because of the power that you actually give, the frequency at which vibration is carried out. And as a result, usually around 700 base pairs or 600, 700 base pair long DNA fragments can be generated. It can be optimized based on the machine you have. And then those fragments can be used for sequencing purposes. So that is the idea that shotgun that I just mentioned is usually focused on. So the first step, if you look here for both of these techniques, is that from the intact DNA that you initially have, you have to subject it to fragmentation or you have to break it down to make it easy to, sorry, this is hurting. Yeah, to make it easy to study further. Next, what happens is that this genomic DNA for shotgun Sanger sequencing, the genomic DNA after fragmentation is cloned into a vector of your choice, and then it is used to transform into E. coli. So I'm assuming if you are already familiar with basic microbiology techniques, it would be easy for you. But in case you're not, the idea of transformation is that any DNA of your interest, if you want to get a large amount of that DNA, or if you want to easily transport it from one place to another, one of the ways in which it is done, it's not the only way, one of the ways in which it is done is by introducing it into bacteria. And then bacteria will act as the host for it. And this, of course, only works if the DNA is present as a plasmid. I'm only talking about plasmids here. Then that plasmid can be used or it can be amplified in bacteria. Why? Because bacteria grows exponentially, very, very fast. And as a result, you can get a large amount of DNA from a very initially very small amount by the process of transformation. So we take any DNA of our interest. Say, for example, I have a vector which overexpresses my gene of interest at very high levels. And currently I have it present. I have a very small amount of it and I need like micrograms of that particular plasmid. So for amplifying it or for increasing it a number, what I can do is I can use a very small amount, 50 to 100 nanograms of the DNA and introduce it to a very small amount of bacteria, competent cells. Competent cells are those which have been prepared to take up DNA. How that preparation is done, etc., all of that you can study in your own time. Basically, these are have been treated in such a way that their membrane has become such that they are very receptive to the introduction of DNA. They do not reject it. They do not recognize it as foreign and chop it up. And they act as very good hosts that can easily take up DNA. So your plasmid of interest can be introduced inside bacterial cells, and then those bacterial cells can be allowed to grow. And when they do, they replicate because you put some sort of an antibiotic selection marker, like ampicillin or kanamycin, one of these drugs, and you allow bacteria to grow, sorry, to grow in a medium which contains these antibiotics. Then under that drug pressure, these bacteria are able to replicate your DNA of interest and they're able to create colonies. And from that colony, you can isolate much more amount of DNA. You can use it to set up bigger cultures, midi prep, maxi prep. These are all these methods which allows you to get lots and lots of DNA that you can use further for your experimental procedures. Now, in context of sequencing, what is done in shotgun Sanger sequencing is that after cloning is performed, it is amplified in bacteria. And for each sequencing, I'm reading the legend here currently. For each sequencing reaction, a single bacterial colony can be picked and plasmid DNA can then be isolated. Each cycle sequencing reaction takes place within a microliter scale volume. So within a very small volume, the reaction actually takes place and it generates a ladder of DDNTP terminated dye-labeled products, which can then be subject to high resolution electrophoretic separation within some capillaries of the sequencing instrument. So here is what you see for cycle sequencing. This is your template. These are your primers. The CTGAT is your primer. And and then you have a ladder of DNA molecules based on where chain termination has actually occurred. And when fluorescently labeled fragments of these discrete sizes are able to pass the detector, the emission spectrum is used to generate a sequencing trait. So overall, the idea of Sanger is remaining the same. Where it is going different is that um, you are introducing your DNA into bacterial cells, expanding it, and then subjecting it to Sanger sequencing. However, in shotgun sequencing with cycle array methods, common adapters are ligated to fragmented genomic DNA. So this is your fragmented genomic DNA, like we just mentioned. And to that, you have
Why
adapter
are we
ligation being,
using this process?
sorry.
Why are we using this process? Because in this, what I can understand is we are replicating this. Like we are making copies of our DNA. So why can't like is it used in like it is used previously or we are using it now also? Like
The
we can use
copies
PCR
of
for
DNA
that.
thing, yeah. So what you're saying, the copies of DNA that is made in third generation sequencing. We'll come to that next. So initially, what was done in the third generation sequencing, the first step only is different. That there you make copies, there template amplification is done because one molecule of DNA is not going to give you complete information about a region of a genome. You need millions of copies of a particular region, and that will give you confidence that it is not an error, it's not an artifact, it is with so much depth and so many number of reads are giving you confidence that some position in the genome has this exact base. And why is that necessary? When you compare it with any disease, you will have confidence that having a change in that region is so incorrect and is a major contributor to the cause of that disease. So that strategy is different. In second generation technique, which is what I'm talking about here, amplification is not done in that manner. Here, you just need sufficient DNA. You need DNA to be increased in a manner that you can carry out multiple amplification strategies. Multiple PCR reactions or multiple uh, sequencing reactions. So this is still limited. Third generation is much larger. Now, please raise your question because I think I interrupted you.
Uh, Ma'am, like I was like curious that why we are using like this vector phenomena is very like tedious process. So
Yeah,
why
yeah,
can't we
these are
just
all,
because
these are all backdated techniques. It's not something that
yes,
you
yes,
do
that
now.
is what my
Yeah,
question.
yeah, yeah.
Okay, okay.
Second generation is earlier on. Currently, Sanger that you do, preferably what is done is either, so yeah, again, it's when I say backdated, it's not completely obsolete. So like I'll tell you from experience, I got something Sanger sequenced a while back. So the company gives you options. Either you can completely purify your DNA in a large amount and then give them the DNA. They will simply sequence it using two sets of primers. That is one way. Second way to do it, when I say primers, it's like imagine this is your plasmid. They will use one primer from one end, and you have to give your primer from one end and another primer from another end, and they'll keep on sequencing it from both directions, right? So when this sequencing is performed, the overlapping region will confirm that yes, your sequence of interest is present in both because your sense strand and your antisense strands are getting confirmed. This is one way. Another way is you can give a large amount of plasmid. What they will do is they'll use primers to PCR purify your sequence of interest. So they will use primers which only select out your region of interest and then they will only sequence that amount. That is another way. Another option, you can give them bacterial stap cultures. So what you are giving is a population of bacteria which are carrying that DNA of interest. They will purify that plasmid and they will take it forward. So there are multiple ways in which this can be done. So while these are backdated, again, they're not obsolete. Um, it's see if somebody has an old sequencing apparatus in their institute, they can use it. Why spend millions trying to sequence when you can just get it done? So it's somewhere in the middle. It's happened over evolution. And also the articles that I've taken this from, they're quite old. It's from 2008. So why I took it is because they have the basics quite clear, but it's not very relevant in today's time. So
So,
yeah.
ma'am, after this like after getting these number of copies, they are using restriction enzymes for getting the desired like the sequence which we have added in this. plasmid.
Where are you asking for the second generation for Sanger or for this one?
Yes, ma'am. Like for Sanger, ma'am, like they have why they have incorporated this to increase the copy, and to
Hmm.
increase the copy there is also a like DNA of bacteria as well, like plasmid DNA is also incorporated with
Hmm.
that DNA.
Hmm.
So
Correct.
to isolate our desired number, like our DNA, they are using
Hmm.
again they are using restriction enzymes, and then they are getting our pure like
No,
pure
no, no.
DNA.
Um, no. So what they're doing is, see, from, say for example, from this entire genome they have, just as an example, they have 50 sequences. So each of these 50 fragments, each of the 50 fragments are closed into 50 vectors, and they are allowed to grow. You carry out transformation and you grow them. So then what happens, if on the plate, the LB plate that you have, if there are say 10 colonies, then for each fragment you have 10 different regions from which sequencing will be performed. So they are replicates of the same sequence. How that will help is because from each of these colonies, you will pick up the DNA, you will isolate it, and you will carry out the same set of reactions. So every reaction that you do will gain more confidence by more number of replicates that you're getting. If you took only one colony and carried out sequencing, it might have been wrong. So for that purpose, they are doing this amplification. Of course, it's laborious. In today's time, we don't resort to this, and the restriction digestion thing that you're saying, it actually doesn't happen here. Here they just carry out cloning, and these regions are sequenced, and then you can use primers to cut it out also. There are multiple ways of doing it. I don't think this is dependent on restriction enzymes. No. You can use sequencing reaction. So because you know the sequence of your plasmid, right? So you can use adapter sequences or primers close to the insert that you have used, and from there sequencing will take place. So the rest of the backbone is not required only. It's just there to complete a plasmid. So could I explain it or should I try again?
No, no, ma'am, it is clear. The second, like the B portion is not clear. The first portion is clear.
Okay, so the B portion is for, yeah, I think I didn't go there. Yeah. So for the second method, that is also shotgun sequencing. Again, the shotgun term is because of the way in which DNA fragmentation is carried out. Now, what is happening to it is that you are ligating adapters. This is the first time the adapter concept is coming in our class. So adapters are ligated to this fragmented genomic DNA, and then it is subjected to protocols that results in the formation of polonies. Polonies are PCR colonies. It's just nomenclature, simply put. So adapters are, how adapter synthesis occurs is again another thing to read about. It's more proprietary in nature, but basically the idea is these are known sequences that you are ligating to an unknown sequence so that you have a starting point from where you will now decode what is that unknown region. So adapters are these light blue or sort of turquoise colored small regions which are getting attached to your region of interest, right? Next, what is happening is that there are small regions within which PCR will be carried out. How is that done? Let's read it. So each polony, it consists of many copies of a single shotgun library fragment. Example being this blue colored fragment. Okay. What happens is that they are all tethered to a planar array. Planar array simply means think of it like a slide, a simple slide, within which there are small regions where you have a gel. What that gel does, again, I'm giving you examples of the most like initial strategies that were used. These are not used in today's time, but a gel basically restricts the movement of molecules out of that region. So within a very small region you are forcing the sequencing reaction and the PCR reaction to occur. So inside that region, what all is present? First is your template. That template is this blue colored fragment. Second, what is present? You have multiple reaction volumes. You have your primer, you have your enzyme which will carry out polymerization, etc. So what this idea of Polony basically does is that it is restricting this region, and within a microliter scale reagent volume, you will have primer hybridization, you will have enzymes which will be able to carry out extension, and then you can actually image it. So what it does is within a small region, imagine a slide, you have multiple small colonies, and within each colony, independent sequencing reactions or amplification reactions and sequencing reactions are taking place, and they are also fluorescently labeled, as a result of which each time a base is getting incorporated, you will be able to detect it. So imaging based detection of these fluorescent labels, sorry, incorporated within each extension can be used to acquire sequencing data of all the features in parallel. So what this is giving you as an advantage is that in one snapshot, every time, every few microseconds, for example, if an image is taken of an entire slide, you will be able to get the data for each of the colonies. So how do I explain this better? You will see the application of this technique later on, that currently this imaging based quantification is still being done. But basically the idea is that you want to get more information in 1 second, in one snapshot. So here you will have, say for example, this snapshot shows you what is base 1. So for if all the bases, if all the polynase are starting their sequencing reaction at time x, and you take a first snapshot at time x plus 1, then you will know that which is the first base that got incorporated in all those fragments at the same time. Again, what is base 2? Again, what is base 3? And so on and so forth. So every few seconds, you will get some, every few microseconds, you will get some information, and you'll be able to see what is the base that is getting incorporated. This will get more clear once I explain further techniques. Don't worry about it. But basically, the idea of Polony is something that has been modified and has been more made more efficient. Previously, what was done using gels, people tried to localize the sequencing reaction in a very small region. Currently, companies have like commercialized it and made it huge, and they have taken up multiple strategies to make it very proprietary also. So you can't copy those ideas, and we don't know those techniques in a lot of detail. But whatever is publicly available, I will show it to you. There are videos also which will explain it. So basically, what is the difference? Compared to shotgun Sanger sequencing, Sanger sequencing was still dependent on the incorporation of labeled ddNTPs, like we already know. And every time a ddNTP got incorporated, fluorescence-based detection can be carried out. And therefore, you will know that at this position, this particular base is present. In comparison, when we talk about second generation sequencing, which is based on cyclic array methods, then every time a base keeps on getting incorporated, you will be able to detect it using a fluorescence detector that will know that this base has gotten incorporated, because you have 4 different fluorophores for 4 different bases. So it is an imaging based, more high throughput technique for shortcut sequencing. So you'll learn more about it. Just hold a little more patience. I'll explain everything in a lot more detail. So yeah, here is where we go into more detail about what do we mean by second generation. So it is usually used in reference to various implementation of the cyclic array sequencing that has been realized in commercial products like Roche 454 sequencing, so Alexa technology used by Illumina. This is what we'll be discussing in extensive detail today. Because Illumina sequencing is most commonly used, most reliable, and in most number of studies, that's what you'll find. Then there's the solid platform also, and the Heliscope single molecule sequencer technology. Now keep one thing in mind, that cyclic array sequencing is a strategy. Each of these companies and their instruments have modified them in ways which makes them their intellectual property, and a large amount of information by these companies is not disclosed, but the basic idea is disclosed, and that is what we will discuss today. Okay, so what do we mean by that term that I just explained? Cyclic array sequencing. So summarized, it can be called as the sequencing of a dense array of DNA features by iterative cycles of enzymatic manipulation and image-based data collection. Lot of jargon. What does it mean? First, you have a very restricted region from which you are collecting information. Second, you have multiple cycles where a reaction is taking place that is going to give you incremental data. So every cycle is giving you some data. Third, how do you detect that data or how do you understand that data? You use some imaging based data collection method. These are the three properties. How does it happen? What is going on? You will learn it gradually. Again, hold some patience. So, yeah, one of the ways in which this restriction that I was talking about, that trying to limit the sequencing reaction within a very small space is actually done, is by using emulsion PCR. So what does it mean and who actually uses it? So if you remember here, I showed you, mentioned all of the commercialization of these techniques, which has been done by these companies like Roche and by Solid, which is by Applied Biosystems. So these platforms, they rely on this emulsion PCR method. This is, I think till date, this is what is being used even now. What do they do under this technique? In brief, in vitro constructed adapter flanked shotgun library is PCR amplified. What does it mean? It means here you have a shotgun library. How is that generated? We already know, by fragmenting DNA. Fragmentation is performed not by restriction digestion, but it is a random fragmentation of the entire genome so that there is no bias, and you get a representation of all regions within the genome so that whatever conclusions you make are very correct. So to that fragmented DNA, we attach these small regions of known sequence that can be used for um, further further sequencing reaction, and those regions are called as adapters. So you have adapters for 5' regions and for 3' regions, which are shown in yellow or gold and turquoise. What do? They each are flanking unique inserts. Each of these four inserts, you can see they are differently colored and they are all flanked by these adapters. What happens to them? They are PCR amplified. PCR amplified, again, why? Like you just mentioned, template amplification is a major step and it is necessary because whatever techniques we're discussing, they are for high throughput data. Now, PCR amplification that we are talking about is multi-template PCR. It is not multiplex PCR. There's a difference between both of these. It means that multiple PCR reactions are using the same template. But for multiplex PCR, you use multiple primers for the same template. Here, we are not trying to do that. We are just using multiple templates for the same purpose. So what do? They are these primers that I'm talking about, they correspond to the gold and turquoise adapters. That is why knowing their sequence is very essential, because otherwise you wouldn't know where to start. One common strategy that is used when we don't know a sequence and we want to be unbiased in amplification is the idea of using random hexamers. But understand that it is going to get incorporated in random sites within your DNA sequence, because it's just a combination of sequences, right? Now, while that suffices when we are trying to do cDNA synthesis, first strand cDNA synthesis, when your objective is to get the entire sequence of a DNA fragment or an RNA molecule, then you don't want abrupt regions to get sequenced. If you have a stretch, you want both the ends to be your anchors, and from the anchors, you want both sides to get sequenced, and you want to overlap those sequences, and you want to get complete confidence that yes, from my 5' end and from my 3' end, the sequencing output that I'm getting are actually corroborating with each other. And therefore, this is the final sequence of my region that I'm interested in. Okay, so that is the idea. Now, this entire step or this entire strategy is carried out within an emulsion. So if you've studied emulsions, you would know that basically you have two different types of liquids that are of different densities, and when they are subjected to agitation, then there can be formation of droplets. Most common, water in oil emulsion are formed. So what it does is it creates these little droplets that you can see, and in each of these droplets, what you have is a bead. These are very small beads. Like you can see, these are micron scale beads, and they are bearing the primer for the adapter that you have already incorporated. So what did we do? We generated fragments of our DNA of interest, and to that DNA, we have ligated adapters. Next, what did we do? We took water in oil, sort of a solution, water and oil solution. So, and we to that added some beads. These are made of different, these are sort of inert beads which will not affect the reaction in any way, but they will just provide like a sort of surface for the reaction to take place. So these beads have immobilized primers. Immobilized as in they have covalently attached primers on the surface, which can bind to your adapter of interest. Now what is done is using a droplet generator. What is done is that the water in oil emulsion is agitated, and as a result, small micro droplets are formed, like you can see here. And it is a chance event or a random event, and then and a productive droplet is formed when you have a reaction where the bead is present containing your primers, you have your enzyme, and you have your template. When all of these individual components are present, only in such a droplet can PCR reaction take place. How is that good, or how is that important? It is because these sequences are so many in number, and because one of the major challenges with amplification or with sequencing is the fact that you get limited by the amount of space, and even if primer is present, primer is not able to bind, or even if your template is present, it is not able to denature. There are a number of physiological interruptions that actually can affect your sequencing reaction. So what people usually try to do in a large number of cases is to give some sort of a support on which the sequencing reaction will take place. In this case, that support is a small bead, a micron scale tiny bead, and on that bead, PCR reaction is actually going to take place. So what does it do? One of the PCR primers, like I just mentioned, is tethered to the surface of micron scale beads. A low template concentration results in most bead containing compartments having either 0 or 1 template molecule present. So you have to titer it accordingly. Titer means you have to give a range of concentrations, and you have to determine what is the most optimal concentration to make sure that one droplet only has one template reaction, and so non-specific results cannot be ascertained. In productive emulsion compartments where both a bead and a template molecule is present, PCR amplicons are captured to the surface of the bead. Now, once within this particular droplet, PCR reaction has been completed, you break the emulsion, and then the beads that bear the amplification products can be selectively enriched. So you can select out only these beads which actually have your amplification products and remove the beads that did not undergo amplification. Each clonally amplified bead will bear on its surface PCR products corresponding to amplification of a single molecule from the template library. So what have you done in this step? You have enriched for your template of interest, and you have beads which are, so on one bead you will have, this is just showing you, I think, around 8 molecules, but imagine hundreds and thousands of such molecules present on the surface of one bead. So you are enriching the availability of the template in this strategy using emulsion PCR. This is one strategy. Second, you have also have to try library preparation, which is accomplished by random fragmentation of DNA followed by in vitro ligation of common adapter sequences, which is what we have discussed as well. The generation of clonally clustered amplicons to serve as sequencing features can be achieved in multiple approaches. First being in situ Polynase, like I mentioned, which is not very commonly used in today's time. You can use emulsion PCR, which is the second strategy that I just mentioned. And third is the Illumina bridge PCR. This is what we will discuss in more detail. But if you have doubts from the emulsion PCR part, please ask before I can proceed. Let me know if there's any query, anything that was confusing, just let me know.
Ma'am, after attaching like our DNA fragment to the beads, then we are performing like uh, replication, and then we are uh, getting our uh, sequence. Is it true?
Yeah. The sequencing part comes later. This is the initial part. So um, let me go back to this slide. That will, okay, I think the title is what might be confusing because I've kept key concepts as the title. Maybe I'll change that. Just a second, my system is just not moving. Yeah. So if you look at here, the first line here talks about the NGS workflow, which has sample extraction, library preparation, sequencing, and analysis. We are talking about library preparation today, majorly because that is a huge concept just by itself. And sequencing overall, you know the idea of it. You just want to incorporate bases and you want to get a readout of the base. It's not as simple as I'm mentioning it. That also has a lot of variation, a lot of companies doing different things. But currently what we're discussing in more detail is this library preparation. So as part of it, you have this concept of creating clonal clusters as in a dense region where same type of DNA is present, so that from that region, whatever readout you get will be an amplified fluorescent signal that can be detected using imaging based techniques. So a snap. So for example, imagine if you've read microarray, this would actually be easy to understand. But like if you have one DNA molecule on one chip and that one DNA molecule is getting amplified, the amount of signal you'll get is very small. So for signal amplification, you want to cluster, say 1000 such DNA molecules, just as an example. And when from all of those you get the same signal, you get the same red signal or green signal, then that becomes very easily detectable. And then that also has more confidence. So that can be interpreted as the correct result. So that is what happens. So as part of library preparation, we are discussing the different ways in which different companies do this. That Polynice strategy that I mentioned, that's not, as you can understand, it's unnecessarily laborious to some extent, but I think it can be done. It's of course a feasible technique, but it's just very for a company, it's not very practical because it will take a lot of time, and the cost effectiveness of it is doubtful. The second one that I talked about, emulsion PCR, it is done. It is done by all these companies in today's time, and it is also a strategy which is used for droplet digital PCR as well, ddPCR. When if your template is something that is very limiting, um, normally for qPCR already we are able to use very little amount of template and then detect the expression of a gene. But when you have even lower amounts, then people generate these small droplets, and within that droplet they detect the presence of a particular RNA, and then they conclude that yes, it is. It's usually done for organoids and any sort of condition which is from very small sample, from patient tissue, very crucial tissue. All this is done. So emulsion PCR is another strategy, and the third one that we'll talk about is the one which is which was a huge unbelievable step that was taken forward by Illumina, and it is called as the bridge amplification strategy. So that's what we'll discuss. But I hope you understood your query. We are only talking about library preparation for now.
Yes, ma'am.
Okay. Okay. So I also have a video to explain this much better. Maybe we can, yeah, maybe we can start off with the video. And I'll keep explaining along with the video because it's a company's it is the company's own video to explain the bridge amplification strategy. So you can watch it on YouTube for your own understanding. It's just not ending. Give me a minute. So
the Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing.
yeah, so these are the 4 steps that I've already mentioned.
There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments.
So like you can see, this is your DNA sequence of interest, and middle in white, and you attach 5' and 3' adapters to the same to prepare your sample. Next,
through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
okay, this is information that we have not already discussed, but basically think of them as additional markers for you to know where exactly or which region you are sequencing, which molecule you are sequencing, and you're also attaching certain regions which will be able to bind to the solid support that you would, like in case of emulsion PCR, you had a bead on which you had primers. In this case, primers are present on a different sort of a surface, which is called as a flow cell. You will learn more about it subsequently. But on the flow cell, you will have these short oligonucleotides to which your sequence of interest or your fragment of interest will be able to bind only when this adapter ligation has been completed. We'll learn more about it. Let's see ahead. So this is a
Clustering
flow cell.
is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands.
So this is why it is extremely crucial to do the first step that we talked about when we were discussing library preparation. So you have all of the fragments that you have, they need to be attached to or they need to be ligated to the adapter, because otherwise they will not hybridize anywhere. And even if your sequence is present, it will go unrecognized. And any information you conclude from such a sequencing analysis will be A, incorrect, and B, possibly inconclusive as well. So adapter ligation is very crucial. And as you can see, the flow cell has simply, it was like a slide, like I just mentioned. So on that slide, it has a representation, and it's not a very continuous distribution, but it has overall same or similar number of both types of the adapters. When I say types of adapters, I mean sequences which are complementary to the adapter. That is why knowing that sequence is important. It helps you recognize that this is where my sequence should bind. Now,
a polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand...
So I hope you understood this part. So let me just, yeah. So from this, yeah. So focus here, what is attached to the base of the flow cell is your adapter, and to it, your sequence of interest bound, that was the template, and complementary to it, synthesis was carried out by the polymerase, right? Like the question
Polymerase
here.
creates a complement of the hybridized fragment.
So after this region has been polymerized, your initial template, what is the template? Your template is the fragment, DNA fragment, which you have adapter ligated and added to this reaction. That is removed. How is that going to help? That is going to help because this can again bind to somewhere else on the flow cell, some other region, and this can keep on amplifying your signal exponentially. It will do this n number of times, and it will keep on giving you more and more information that you can, gain more confidence into.
Okay. The strands are clonally amplified through bridge amplification.
Now we come to bridge
In this
amplification.
process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell.
So the whole concept of bridge amplification comes from exactly this, that the molecule that was initially synthesized as using our DNA of interest as the template can fold over, bind to the neighboring oligo, which is complementary to the other end of the DNA, and then PCR reaction can again take place. So there are two polymerization reactions which are taking place, and now these will remain bound to your flow cell while the other one, which is the template, is getting washed away.
The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce.
Okay, so till now, library preparation has been completed. We have not yet moved into sequencing, like I just mentioned, but I hope that part was clear. Moving on to how the actual sequencing process takes place.
The first read.
So, just
Priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition,
if you noticed here, every time a base got incorporated, the corresponding signal was also emitted as a fluorescence. So the blue tag, which is attached to all the ATP molecules, DATP molecules, are emitting a blue fluorescence. So that can be detected at the time of imaging by the fluorescence detector.
After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis.
We will learn more about this. So again, this is proprietary because it was Illumina's concept. So basically, sequencing by synthesis means that you are synthesizing the you're using a template strand and you're synthesizing the complementary strand. And at the time of synthesis, you are also decoding the sequence of that molecule. So therefore, the concept of sequencing by synthesis was kind of proprietized by Illumina. So
synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call.
These are all the different attributes which are taken care of when you get your sequencing report. You don't have to worry too much about it, but basically what it is saying that how many cycles you are actually doing for the same is telling you how long the read can be. So commonly when we get sequencing done in today's time, it is around 100 base pairs. Initially it used to be around 36 to 40 base pairs. So it has increased a lot today. And also we often go for paired end reads, which means for the same read for the same fragment, you get sequencing done from both ends. So you get more confidence in your data for RNA-seq.
The emission wavelength, along with the signal intensity, determines the base.
So the base call basically gives you more confidence that for this position, when this base was incorporated, the amount of fluorescence intensity which was given out as the emission wavelength, it sort of gives you confidence that the incorporation was correct. So it is a jargon, it's a terminology, which is base call. That report also you get.
Called. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the.
Okay, let me interrupt you here. So, basically, till now, what we saw is there's a lot of information because they have taken it to, I think there's only a little left. We'll go to the end of the video, but they have moved on till data analysis part. But we were till now kind of stuck in the library preparation part only. But just to like give you a teaser of what happens is that think of it logically. Let me try to switch on the video, it might help. So think of it in this manner that if you have sequenced so much information, you have like a number of DNA molecules that you have sequenced. Now, how will you know where that sequence came from? Because what you have actually sequenced is a fragment. You should be able to figure out which region of the genome it came from, what it represents, and how many times that particular region of the genome got sequenced, so that you can figure out what is the depth of sequencing, what is the confidence with which you can be sure that this region has undergone sequencing correctly, and whatever base I'm getting is true. It's not a fallacy. It's not a polymerase error, or it's not an any incorrect information. So for that, indexing is done. This indexing is very similar to the indexing you do in your copy, right? So it's like it's imagine it to be similar to putting a tag as to where it is present, and that indexing is necessary so that you know where your data has come from. Like you have um, so many bases and so much information in your hand. Now to make sense of that information, indexing is necessary, and that is where that is most relevant when you're doing data analysis. We are not going into it right now, so don't worry. It'll come maybe in the next to next class when we learn more about data analysis and what is required. But basically, what you did was you got initially took the sequence of the send strand. You washed off the other strand, you used index primers and got the rest of the sequence. Then you again got another, you took the other strand and took the reverse strand sequence also, washed it off, and now you have information from both these strands, again giving you more confidence. So a second, it's getting started. Yeah.
Sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignment.
Yeah, so I think it got a little stuck here. But basically, what they're saying is that after you have all of that information, you need to make sense of it. And to do that, you have to stitch together all of the sequences that you have gotten. And for that, indexing is very important. Once you have indexed that this has come from this region of the genome, you will take all of the reads that have come from neighboring regions and you will stick them together. So if you have a long sequence from there, you will have read in region X, region Y, and the region between X and Y as well. So when you stitch all these little fragments together and you align them to the reference genome, you will be conclusively able to infer that yes, this is the order of the sequences. And particularly this step is very crucial because any errors in this step can lead to very incorrect results. Just excuse me for a second. Yeah, sorry for the interruption. But basically, this is necessary for aligning to the reference genome.
The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in base space sequence.
So this is just advertisement, but basically this is the sequence by synthesis workflow. If there's any doubt from this part, you can ask me and then we'll move back to the presentation. Any doubts?
No, ma'am, it is clear. I have watched that video.
You have? Okay, yeah, it's a very, very informative video, gives you a lot of information. So, yeah, coming back here, we don't have a lot of information. Let's say 30. Okay, we'll wrap it up in 10-15 minutes. So, so basically, this is what Solexa. Solexa is like the initial company name for Illumina. So what they do is they rely on this bridge PCR technique, like I just mentioned, which is clustering. So what it does is it amplifies certain regions in a to generate clusters of clones. In brief, you have an in vitro constructed adapter flanked shotgun library, like we just understood, which undergoes PCR amplification. But both the primers, they coat the surface of the flow cell, like we just talked about, and they are attached to the flow cell using a flexible linker. As a consequence, any amplification products which originate from any given member, let me just get the uh, any of the amplification products which originate from any given member of the template library, they remain locally tethered near the point of origin. At the end of PCR, each clonal cluster, it contains thousands of copies of a single member of the template library. Accurate measurement of the concentration is crucial to maximize cluster density. That's again not something you need to worry about. You just need to conceptually know about it. But of course, it's the company that takes care, and the experts, technical experts who take care of it. So that is the idea. Now, just a little bit of an insight into the chemistry that goes on here. We understood the sequential steps, the technical details of it, but what happens from a chemistry point of view? So on the Illumina platform, the bridge PCR is somewhat unconventional because it relies on alternating cycles of extension with BST polymerase and denaturation with formamide. The cluster that we talked about, we said it consists of nearly 1000 clonal amplicons. Again, please keep in mind, this is also an older version of the done a lot of updating, and not all the information is disclosed, like I previously mentioned. So you can check it once again for more information. But what you can see now is that what is generated are millions of clusters that can be amplified to distinguishable locations on a single flow cell that can all be sequenced in parallel using the same instrument. So you don't have any of those external variables that you use different instruments at the for sequencing different regions, or that you used maybe the efficiency of the instrument was not the same at that point of time. So all those variables are removed. You are carrying out parallel sequencing of multiple regions. So after cluster generation that we talked about, the amplicons, they are linearized and a sequencing primer is hybridized. So I think, yeah, this we can use as a reference. So this is your to the flow cell. These are your bases to which it is amplified, and from to which it is tethered. And from there you have sequencing being carried out using the adapter. It undergoes ligation, and from there what you do, you image the array. Then these two terminologies are something we have not discussed: a label and a terminator. What do we mean by that? So the nucleotides which get incorporated into or during this polymerase reaction, they are modified, chemically modified in two ways. What are they called as? They're called as reversible terminators. How so? A chemically cleavable moiety at the 3' OH position allows not only single base incorporation to occur in each cycle. So if you think about it, the concept is quite similar to that of Sanger sequencing, where you used a DDNTP to make sure that chain termination occurs right there. Similar to that, here also you are ensuring that only a single base is incorporated and extended polymerization or extension cannot take place. So a chemically modified base is incorporated, and each of these bases obviously also carries a fluorescent label which is chemically cleavable. So that gives identity to each of the nucleotides. So after the base has been incorporated, there is the removal of the of the modified of the modifying region, like the chemically modifiable region, and the fluorescence label is also removed, and then again the next base can be added. So this is necessary to make sure that in tandem you don't have 5 6 bases getting incorporated one after the other and their signals keep on getting mixed. So all that is of course optimized. So after imaging, you remove the label, you remove the terminating moiety, you add fresh nucleotides, you add polymerase, and again the reaction can proceed further. So that's how it keeps on getting repeated. Next, yeah, so that was the overall idea. You can gain more idea, you can gain more insight into the chemistry of it. What are the molecules that are used? What is the chemical definition of the same? All of that you can also look at. I think we might have it in one of the slides. Let's see if we can like complete that also by today. But for now, to summarize the template amplification strategies that I talked about, the first one is emulsion PCR, majorly used by Roche solid sequencing and QIAGEN and Ion Torrent as well. So here you have one tube where you have these emulsions within which you can see there are these tiny beads on the surface of which you have adapter complementary primers present. They have the template, they have the primer, they have the NTPs and polymerase. Amplification is carried out on the bead. So templates are hybridizing to the bead bound primers and they are amplified. Following that, the complementary strand dissociates, leaving bead bound single stranded templates. These 100 200 million beads with thousands of bound templates are then used for sequencing purposes. So that's what is present here. You have this aqueous droplet, which is a micelle, which has a bead with these other components, and PCR is carried out within the micelle, and it covers each bead with thousands of copies of the same sequence, which can then be enriched and then be used for sequencing. In comparison, when we talk about solid phase bridge amplification, there already, like we have discussed, you have templates which are binding to slide bound adapter sequences. Bridge amplification taking place where they bend over and they are able to carry out polymerization reactions. Distal ends of the hybridized templates interact with nearby primers and then amplification can take place. And finally, cluster generation takes place where 100 to 200 million clusters, clonal clusters are formed. This is a pattern flow cell which increases the cluster density and enriches the signal for a particular region. So yeah, that is overall the idea used for Illumina. Then you have a concept of solid phase template walking. Oh, just a minute. I'll be yeah. Okay. So yeah, so for the solid phase template walking that I was mentioning, for that fragmented DNA is ligated to adapters, like you can see here, which is bound, and it is bound to a complementary primer, again attached to a solid support. Here also PCR is used to generate the second strand, and the now double-stranded template is partially denatured, allowing the free end of the original template to drift and bind to another nearby primer sequence. Uh, Quite similar to what you see in Illumina as well. Major difference being the fact that bridge amplification is not occurring. So you don't have two types of primers being present on the same surface. And so amplification is, the strategy for template amplification is different. Now, reverse primers are used to initiate strand displacement to generate additional templates, each of which can bind a new primer. So let's look at the image for more clarification. You have the solid support on which your primer is present, to which DNA template is bound. The second strand gets amplified. For primer walking, dsDNA is partially denatured, allowing the free end to hybridize to another nearby primer. Then template regeneration takes place where more and more template amplification is occurring. Free DNA templates can be generated, they'll be washed off, and after several cycles, clusters are present on a patterned flow cell, which increases the efficiency of signal amplification. So this is, again, like I said, quite similar in a lot of aspects to solid phase Illumina strategy, only major difference being the bridge amplification method for Illumina. Finally, in the fourth strategy, which is used by Complete Genomics, another company, they use in solution DNA nanoball generation. So this is a completely different strategy, and it's something that we don't very commonly hear about as well. But I'll try to explain as much as I can. So here in DNA nanoball generation, first, what is done? DNA fragmentation is carried out like we normally do, followed by which you have ligation of an adapter. What is the key difference here? This will not be the only adapter which will be ligated to the DNA in the entire course of time. Instead, there are four adapters which are used in these sequencing reactions. Only the first one is hybridized to the fragmented DNA, like you can see here in red. Following that, what is done? DNA is template circularization is carried out because the adapters are able to ligate to each other, and then you have a circularized DNA. Then it is cleaved with a type 2 endonuclease. Because it's a double-stranded DNA, it is able to cleave double-stranded DNA. Restriction endonucleases are able to do so, and that cleavage is done downstream of the adapter sequence naturally. Following that, what will you do? You will add another adapter molecule to these ends. Like basically, if you cut this DNA, again, it can get linearized. You can again incorporate two new adapters, which can again circularize. Then again, that DNA can be cut, and you can generate another site where another set of adapters can be ligated, and so on. Similarly, iterative ligation is carried out, and following that, you will have a circular template where four different adapter sequences are present, like you can see here in red, blue, green, and yellow or orange. So what it is giving you is a complete circular template, which will now be subjected to rolling circle amplification. So if you see here, circular templates, they undergo amplification to generate long concatemers called as DNA nanoballs. Intermolecular interactions keep the nanoballs cohesive and separate in solution. Again, this is something that I don't have a lot of insight on, but basically what it does is that for it will keep on undergoing amplification because um, if you have a circularized molecule and if you have one single nick from which point it will use the other strand as the template, it will keep on continuing in a concatenaric state. And each of these nanoballs can then be immobilized on a patterned flow cell, uh, which is then subject to sequencing. So it is another very different method of template amplification. It is just giving you more regions for identification of the or rather for starting the sequencing reaction because multiple adapters are present. But yeah, it's a less explored, less commonly utilized strategy, but it is something which is done. If you want more insights into it, I can read about it and come back. But again, it's not very commonly used, so we are also not very familiar with it. But if it is something that is interesting, I can certainly give you more material regarding the same. So just the time. Okay, so we'll just wrap it up pretty quickly because yeah, we'll just go through sequencing by synthesis and sequencing by ligation techniques, and that'll sorry, complete the topics for today's class. So like we had previously mentioned, Illumina strategy uses the SBS method, which is the sequencing by synthesis method. think it's stuck here. Yeah, I think it's working. So, so the method, as the name obviously implies, you are sequencing your template DNA of interest while synthesizing the complementary strand, or while synthesis, the sequence is also getting decoded. So, in contrast, when you talk about sequencing by ligation method, it is different. It sequences short stretches and then it connects them together by ligation and then decodes what is the sequence within that region. So the example of this is solid sequencing, again, something we have already discussed, which relies upon emulsion PCR. So following cluster generation or bead deposition onto a slide, the fragments are sequenced by ligation. How does it do so? You have fluorophore labeled 2-base encoded probes. Here you can see the probe is represented here in blue, uh, which is composed of known nucleotides in the first and second position. These two are the known nucleotides, followed by 5 to 6 bases, which are a random set of unknown nucleotides, which are then tagged with a fluorophore. So this entire molecule is your probe, from which your first 2 bases are known, the next few bases are not, and then it is connected to a fluorophore. Keep in mind that the blue corresponding here is representing the known base color, and the fluorophore also represents the same. If you will see here, other combinations of dinucleotides are showing different colors, and therefore the corresponding fluorophore is different. This is important because that actually dictates the identity of the bases which got incorporated. So coming back here, you have a probe which is composed of known nucleotides in the first and second positions, followed by degenerate or universal bases, which is represented in pink, that is added to the DNA library. The 2-base probe is ligated onto an anchor shown here in light purple, the anchor here. That is complementary to an adapter. Again, you know that the adapter is attached to your fragment of interest. And then the slide is imaged to identify what are the first 2 bases that got incorporated. So coming back, what do you have? You have a DNA sequence which has adapter ligation done on both ends. You denature it, you take one as your template. Complementary to the adapter you attached, you have an anchor sequence. So that is fixing your DNA in its position. Next, what you do, you sequence the first 2 bases by using probes which have a set combination that if AT is getting incorporated, a blue signal will be given. If CG is getting incorporated, a light green signal will be given out. So all these different combinations can be used. And based on that, what you can do is you can image the slide and then get an idea of what bases are incorporated. Unextended strands are capped by unlabeled probe or by phosphatase to maintain cycle synchronization. Finally, the terminal degenerate base and the fluorophore are cleaved off the probe, leaving a 5-base pair extended fragment. The degenerate bases are removed and the fluorophore is also cleaved off. This process then has to be repeated multiple times, at least 10 times, until 2 out of every 5 bases get identified. So what do? Look at here, look at this image. It will explain it a little better. What you have here is after this incorporation has been carried out, a cleavage is performed to remove the fluorophore along with several bases so that a 5' phosphate end is exposed. Next, what you can do is probe extension can be carried out where 10 rounds of hybridization, ligation, imaging, and cleavage will be carried out to identify 2 out of every 5 bases. Again, you will find a probe which gets incorporated somewhere here. It will allow only one of the possible combinations to get incorporated. So when GT containing probe is present, it gets incorporated. When AA containing probe is present, it gets incorporated, gives out a yellow signal. Similarly, this will keep on proceeding. So this is your probe extension. Now, from this entire sequence, you have out of every 5 bases, you have 2 bases whose sequence are known. And in between, you have 3 interrupting bases whose sequences are not known. To decode that, what is done? The entire strand is now reset by removing all of the ligated probes, and the process is again repeated using an n plus 1, n plus 2, n plus 3 or an n plus 4 offset anchor. What does it mean? It means that instead of starting from this first two bases, if you now use an n plus 2 offset, then after this initial sequence, the next two bases are also part of the known sequence. So now the probe will start from this position, position number 3 or plus 3 position, where you can see TC has gotten incorporated and that will give out a red signal. Again, the same occurs here. So if you align all of these sequences, what you can see is now you know that GC is followed by TC, you know that GT is followed by AG, and so on and so forth. So the interrupting sequences are now also revealed. So this has to be done with anchors that have an n plus 2, n plus 4, n plus 3, so that all these interfering bases are now also known and the entire sequence can then be decoded. Now, this also is a slightly confusing, but it's a technique that is very commonly used. And of course, solid sequencing is used in today's time also. It has, of course, undergone more upgrades, but the basic biology lies there. Very similar to that, uh, complete genomics uses a sequencing technique where the combinatorial probe anchor ligation method is used, which is CPAL. So if you remember, we talked about the nanoball generation previously. So after nanoball deposition in the flow cell has occurred, you again use an anchor similar to the previous strategy. You use an anchor which is complementary to one of the four adapter sequences. If you remember, we had used four adapters: red, green, blue, and orange. So you use an adapter, you use an anchor complementary to one of them, and a fluorophore labeled probe is bound to each nanoball. The probe is degenerate at all positions, but except the first position. So only one position is known. The rest of it is completely degenerate. So if you look here, the red one is your adapter. You have an anchor complementary to that, and the first base sequence is known. Following that, you have degenerate bases. So this is allowing you to proceed with single base encoded probes. The anchor and probe are then ligated into position and imaged to identify the first base on either 3' or 5' end. Next, this is removed. The process begins again. You use n plus 1 position, n plus 2 position, so on and so forth. And in this way, you keep on continuing it till you have your complete sequence. So for paired end sequencing from the adapter, both left and right side sequencing is performed. So you have information for both the ends. And yeah, so offset anchors, like I previously also explained, n plus 5 offset will include anchor followed by 5 different bases, followed by your known base and degenerate base. So basically that the strategy remains the same. There you had for solid sequencing, you had two known bases. For complete genomics BGI, you have one single base, single nucleotide encoded probe, or you have paired end sequencing followed at both ends. So overall, this is the concept of sequencing by ligation. Contrary to it, sequencing by synthesis is quite different. So it is an Illumina-based, the technique that Illumina uses. We have already discussed it, where you have here a little more insight into the chemistry is given. I think I mentioned it once before. But basically what happens here is that the nucleotide has a blocking moiety, which I mentioned, the 3' O azidomethyl group, and it has a cleavable fluorophore. So in each round of incorporation, the fragments which are present in each cluster will incorporate just one nucleotide because the 3' end is blocked. Once incorporation has occurred, all the other bases will be washed away and the slide will be imaged by turf microscopy. Basically, it will be imaged. Let's keep it to that. And that will give you an information about which base was incorporated. Then the fluorophore dye is cleaved. 3' OH can also be regenerated using this reagent called TCEP, which is tris 2-carboxyethyl phosphine. And this cycle keeps on going, repeating multiple times as long as the sequencing is proceeding. So overall, these are the three steps. You have nucleotide addition, these modified nucleotides that we called terminally blocked nucleotides, followed by imaging using either a 2-laser or a 4-laser channel. Then you cleave it off and then you repeat the cycle once again. For gene reader, which is a Kaizen-based company. What they do is they perform bead-based template enrichment, like we talked about emulsion PCR previously also. Then what you do, you use a mixture of primers, DNA polymerase, and modified nucleotides to each flow cell. Each nucleotide is blocked. Again, the blocking strategy is continued here. So it has a 3' blocking group, which is a 3' O allyl group. And you have some bases which are labeled with a cleavable fluorophore. After incorporation, imaging is carried out. The dye is cleaved. 3' OH can be regenerated using a reducing agent mixture of palladium and TPPTS. So similar strategies used, just instead of a flow cell substratum, here a bead is used. So, and the blocking agent is different, but idea is quite similar. So each bead in this case will emit a color corresponding to the base which gets incorporated, following which the fluorophore will be cleaved, cycle will be repeated, and this will proceed until complete sequencing can be performed. So yeah, overall, that was the topic I wanted to cover for today. If you have any doubts, please, yeah, we're done in time. If you have any doubts, please let me know. I know I give you a lot of information if you are especially unfamiliar with sequencing methods, but yeah, we needed to get done with it by today. So just let me know your doubts and then we can end today's class.
Ma'am, I wanted to go back to the first slide. Else everything is clear.
First slide for today.
Which, yes, yes, ma'am.
Just a second. Let me. This one?
No, 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 ma'am. Second.
This one?
Yes, ma'am. So, ma'am, in this array, are we we are attaching our adapter to a plate or something in this Polony
Yeah.
array? I'm not
So
like.
okay. So think of it as an intermediate to what we have discussed so extensively till now. So it is similar to that. It's a glass slide only. Just instead of what Illumina has, where you have a linker attaching the adapter, here you will have like mostly what I could find in literature, they're mostly gels, like very dense gels. Instead of having immobilization using a chemical linkage or some sort of covalent linkage, you have gel that is very concentrated and that does not allow movement to occur does not allow the diffusion or the movement of the molecules which are present in it. So inside that gel, sequencing is being carried out. But of course, that has undergone improvement, and now people prefer it on a bead within a micelle, or it is performed using substratum of a flow cell. Even flow cell has undergone so much modification today. But yeah, it's think of it like a flow cell, but a more primitive version.
Understood.
Okay. Any other doubts?
No, no, ma'am. Everything is clear.
Okay, so I'll stop sharing. We will discuss long read sequencing. It's just not working. Long read sequencing and a little more detail into the sequencing part of it. We just briefly mentioned the techniques of sequencing by synthesis and sequencing by ligation today. But ion tolerant sequencing and how real-time sequencing is done, all of that will go in a little more detail. And then we'll talk about the actual utility or the difference basically between genome sequencing, exome sequencing in the next class. And that will be on Friday. And we'll move on to hopefully in the next week, we'll shift to epigenetics so that we can incorporate more techniques. And if there are any more topics that we come up in the next class or so on, we'll incorporate it accordingly. So that is all for today. Thank you so much for joining and being so interactive. It was very nice. We'll have our next class at the same time from 7 to 9. So I hope to see you there. And if any doubts or any materials, anything is required, you can connect with me over email. So thank you. I'll end today's class. Have a good night.
Thank you, ma'am.